We will wait until 8 o'clock before we get started, but if anybody has any questions in the meantime, please let me know. I guess I'll go ahead and do the first attendance word, actually, to reward you guys for being here early. So this is extra credit, not one of the required words, but the first word will be snow, since I'm snowed in. That is the first word for attendance. So not only am I snowed in, but my kids also have a 2 hour delay. So as I'm teaching, all my children are here. So please forgive any noise that you might hear in the background. Well, it's 8 o'clock. Slowly get started. This is going to be just like usual. Of course, we're forced to be online today. No one's doing this on voluntarily, but nonetheless, you'll need to send me the attendance words sometime before 9 a.m. And the attendance words are going to go as they have been for the past week, usually, where I will give you some information and then directly after that, give you a question about that information. So, any questions before we? Okay. Which is a good time to remind you. Of course, if you're here, I'm preaching to the choir, but remember to log into your WVSU Gmail account or Google account before you try to join any meetings like this, because if you're not, it won't let you in automatically, as you just saw. And I don't mind helping you and getting you on, but I might miss it, and that wouldn't be good for you. So, anyway, if there's no questions, let's jump back into chapter 4. So this is where we left off. We were talking about cellular respiration. We had talked about first we did the big overview, um, and then we started going back down or started rediscussing these things. But in this case, we did it in more detail, and going through it in more detail. We finished talking about the first stage, which was glycolysis, and this is where we left off talking about the second stage, which is the citric acid cycle. So everything on the screen here we've already talked about, but this is where I left off. And what we didn't get to see was the picture that basically visualizes all these words right here. So there you go. The input, right? So we have acetic acid coming in. We have ADP, right, and a phosphate, so the dead battery, so to speak. We have these things ready to strip away high energy electrons, right? The acetic acid is attached to the citric acid, excuse me, the acceptor molecule, which then becomes citric acid, hence the name citric acid cycle. And then that thing goes through some processes that we don't even talk about in this course. And when it goes through those processes, what we're doing is we're stripping away those carbons, therefore the carbon dioxide. We are using the energy that we are getting from breaking down this molecule to make ATP from this, from the ADP and the phosphate group. And of course, the big picture here, the more important thing is the fact that we are stripping away these high energy electrons that are going to be taken to the electron transport chain, which is where we make more ATP. So as far as the exam is concerned, you know, the questions would be, which of the three stages produces carbon dioxide? That's the citric acid cycle. Which of these which of the three stages is associated with citric acid cycle? Or never mind. I'm probably not going to ask that one because obviously citric acid cycle, citric acid. There you go. But I might ask which of the three stages is associated with acetic acid. right? And that would be the citric acid cycle. And to remind you, this is why I call it the citric acid cycle instead of calling it the Krebs cycle, which is the other name for it, because I want to make things easier for you on the exam. Whenever you see anything about an acid on this exam, you should think citric acid cycle. So any questions about citric acid cycle before we move forward? All right. Before we move forward, the first word, official word for attendance is you're going to need to tell me how many carbons are in acetic acid. How many carbons are in acetic acid? Here you can see citric acid has 1, 2, 3, 4, 5, 6. This acceptor molecule has 1, 2, 3, 4. So how many carbons are in acetic acid? Also, before I move forward, I should have mentioned this earlier. If you want to, you can look deeper into this for independent work, right? This is a very simplified version. There's actually a lot of steps in between here with this citric acid molecule and this acceptor molecule. Also, there's a name for the acceptor molecule, so you can look all that up. Like, what are the in-between steps? What are the in-between molecules? What enzymes are responsible for each of these steps, right? And we all know, or you know from previously in this chapter, that for the most part, these chemical reactions don't happen without enzymes. So which enzymes are being used here? Anyway, if there's no questions, we'll move forward. All right. So like I've been saying, and I wish we could have had this conversation on Friday because now it's been a whole weekend and you've probably forgotten this, and I don't blame you. But the big picture here, right, is that we're trying to make ATP, right? ATP is the goal of cellular respiration. It's all about making the ATP. That's what our cells need for survival. And as I mentioned when we were talking about the first and the second stages, as far as we're concerned, I think the most important thing that's happening in those first two stages is we're stripping away high energy electrons. And those high energy electrons are then taken to the electron transport chain. They're taken to the third stage. And the third stage is where we make the most ATP. Like, right? That's in a sense, the first two stages kind of just support the third one. So that being said, let's now jump into the third stage. And I'll also remind you, because I forgot to do this today, for the most part, a large portion of this exam, this portion of it anyway, will be me describing something and you tell me what stage, right? So I'll say, what stage is this used? And which stage is this produced? Which stage happens here? Or which stage happens there? So keep that in mind as I'm teaching this, because that's, how the test questions are going to go. Anyway, that being said, let's jump into the electron transport chain / oxidative phosphorylation. And I'll remind you, it's going to be the electron transport chain is what I'm going to call it on the exam. Again, to make things easier for you, because I just want, hopefully that'll help you remember that's all about these high energy electrons. So anyway, during cellular respiration, these electrons kind of fall in a cascade down an energy staircase, meaning I keep using this word high energy electrons, right? I'm not just saying electrons, I'm saying high energy electrons. So what happens is, again, they start off at high energy, but by the end of it, they kind of lose that energy. That energy is released, and that released energy is what we're after. That's what the cells use to make ATP, and that's what we're about to talk about here in the upcoming slides. But if you're familiar with any kind of electronics, you probably know that electrons have to come from somewhere and they have to go somewhere, right? Like when you jump a cable in a car, you use these cables. We have one that's attached to the battery and one that's attached to the ground. Or, yeah, I don't know if you're familiar with electric terms, but anyway, the electrons flow from the battery to the ground. They have to have somewhere to go. They have to have somewhere to come from. That's why if you don't plug both of those in, then you can't jump a car battery, right? They have to both be plugged in. And in a sense, we're kind of dealing with the same thing here, and there will be test questions about this. So even in this situation, electrons have to come from somewhere. They have to go somewhere. And I've already told you where they've come from before, and I've already told you where they've gone. But now I'm specifically singling this concept out. As you're going to learn in these next slides, the high energy electrons come from glucose, and they go to oxygen, right? So that's nothing new. I've said that before, but now I'm saying it and making sure I'm singling out that concept. Electrons are coming from the glucose. Those high energy electrons come from the glucose. They go through all this processes, they get stripped away and brought to the electron transport chain. And we're going to talk about that here in a second. But they have to go somewhere. It's the oxygen that accepts them, right? So they come from the glucose, they go to the oxygen. And when the oxygen accepts them, it becomes, that then gives us a molecule of water. So that being said, let's talk about it. Here's a visualization. Again, electron transport chain is happening inside the mitochondria. And we're finally here in this section of the chapter, we're going to talk about why that's actually important. But again, as a reminder for the test questions, I've got 2 test questions I can see here. Which of the 3 stages happens outside of the mitochondria? Glycolysis. Which of the 3 stages happens inside of the mitochondria? Citric acid cycle and the electron transport chain. So let's talk about it. Again, this overall effect of the electron transport is a downward trip for electrons, so to speak. What I mean by downward is, they start at high energy, and again, by the end of it, they're just, they've lost that energy, they've released it. But again, like I said previously, now here it is in writing, the electrons come from the glucose, right, or other molecules, and we'll talk about that here in a second. But for the exam purposes, we're just going to keep it simple and focus on glucose. So they come from the glucose, they get stripped away by NAD plus, which forms NADH, NAD, yeah. And then also, like I mentioned previously, also FADH2 is one that we learned about for citric acid cycle. And like I said, I'm not going to try to trick you. You don't need to know the difference between them. But if you see anything like this, a molecule that looks like this, you should think, that's the thing that's stripping away and then moving the electrons from one place to another. Anyway, so yes, like I said, they are stripping them away. They're moving them. They're moving them to the electron transport chain, aka the oxidative phosphorylation. And then finally, it's oxygen that accepts it. And when oxygen accepts those electrons, it forms a molecule of water. So there's a lot of test questions there, right? Which of the 3 stages forms water? That would be electron transport chain. Which of the 3 stages uses oxygen? Electron transport chain. Which of the 3 stages strips away high energy electrons? That would be glycolysis and citric acid cycle. Which of the 3 stages uses those high energy electrons that were stripped away? And that would be the electron transport chain / oxidative phosphorylation. So any questions so far? All right. We've got another test question coming. I just gave you this information, so please answer me. Where do the high energy electrons come from in this process? We know that they go to oxygen, which forms a molecule of water, but where do they come from? That is the next word for attendance. All right, I guess we'll move forward. Here's kind of a picture of what I'm talking about. This picture is not from your textbook. So if you're studying this and this picture actually makes a lot of sense to you, then you're going to need to download the PowerPoint or at least access it online, as I'm doing right now. But again, this is just a visualization of everything I said. We're stripping away these high energy electrons. I like this picture. It's a little bit more accurate because it says electrons from food, which is what I hinted at earlier, because we keep saying glucose, and I'm going to test you on glucose. So we just want to keep it simple. But yeah, when all actuality, obviously we eat more than glucose, right? We eat all kinds of different foods, and we break down those molecules and we strip away the high energy electrons, right? So anyway, we get these high energy electrons. They're stripped away by NAD plus, which forms a molecule of NADH. As far as we're concerned, it's basically the same thing, right? This is the empty carrier. This one's full. And so they technically have different names. Those high energy electrons are taken here to the electron transport chain, where they go from protein to protein. And I'm about to talk about this in more detail in the upcoming slides. But yes, they go to different proteins, right? They jump from protein to protein. And as they do that, they're releasing energy. And when they release that energy, these proteins use that energy to make ATP. And we'll talk about that here in the few upcoming slides. But again, like I said previously, those electrons have to go somewhere. Where do they go? They go to oxygen. And when oxygen accepts them, that forms a molecule of water. And you can see here, there's a lot more details on this picture than what I gave you earlier. So technically, when I keep saying electrons, yeah, technically it's 2 electrons at a time. So technically you get 2 electrons that meet up. If this was a chemical equation, a proper chemical equation, you'd have half a molecule of O2. And then that would also form with or meet up with 2 hydrogen ions, these H plus ions, and that gives you 1 molecule of water. But I'm not going to get into that much detail for the exam. So you just need to know High energy electrons, doesn't matter the number of them, are stripped away from food, specifically glucose for the exam. Those high energy electrons, as I'm about to explain here in the next few slides, go from protein to protein, releasing energy. That energy is used to make ATP. Eventually, oxygen is the final acceptor of those electrons. And when it does accept those electrons, that forms a molecule of water. So that's the overview. We're about to go into more detail in the upcoming slides, but are there any questions so far? All right. So like I said, there's the high energy electrons. Once they're taken to the electron transport chain, they go from protein to protein. And there's actually, these proteins are actually built into the inner membrane of the mitochondria. I wish I could draw. I'm not using my usual. Let me see here. I'm using Google instead of Microsoft PowerPoint. Oh, here we go. Excuse me. So this is going to be a really horrible picture. The good picture will be on the next slide. But if this is the mitochondria, there's an outer membrane. So think of it like the cell membrane. It's essentially the same as the cell membrane. There are some differences, right? So you have the outer membrane, like a cell membrane has, but then also there's an additional membrane. And this is a really bad picture because, as you can see on the words on the slide, the inner membrane is actually very folded. So it wouldn't be, I guess technically it would look like this. It'd be very folded up on itself. But anyway, the point is there's an inside membrane, which I just drew, and then there's an outside membrane. And that's actually important. This isn't trivial. Hopefully you know by now that I'm not teaching you trivial things. If I want you to know it, it's because it's not trivial. It's actually important, which is why, for example, when we talked about entropy, for example, I said, you don't need to worry about that. I'm teaching it because it's in the textbook, but just so you know, entropy for us is not important. Well, this is important. Its function, its shape, right? The form of the mitochondria is the reason it can do what it can do. It can do what it does. Excuse me. So let's talk about it a little bit more. Here's a good drawing. Again, here's the outer membrane, right? And then you can see here's the inner membrane. And this is important, as you'll see later, because there's this space in between the membranes. And that space in between the membranes is going to be very important, as you'll see later. Technically, these things are called cristae. I'm not going to ask you that. Technically, this space on the very inside of the mitochondria is called the matrix. I'm not going to ask you that. But yeah, there's also this space in between the membranes, and that will be important when we get there. And hopefully you remember in that previous slide, I said that there's proteins embedded in the inner membrane. You can't really see that in this picture, but just imagine if we had these proteins just right here, embedded in the inner membrane. And that will be important later. And again, to harp on this whole idea of form and function, the reason this is so crumbly, so to speak, it's so folded up on itself, is this inner membrane and is actually a lot larger than the outer membrane. Imagine putting like a big garbage bag inside of a small garbage bag, right? You'd have to crumple it up and squish it in there. And that's exactly what we have here. And that's exactly what we need, though, because again, it's these proteins that are built in to the inner membrane. That's what's doing all the work. So obviously, the bigger this inner membrane is, right, the more surface area you have, then the more proteins you have. And the more proteins you have, the more ATP you're making. And again, ATP is what this is all about. If you forget everything else, remember that, right? So in cellular respiration, the whole idea here is to make ATP. Anyway, any questions so far before we talk a little bit more about how this works? All right, so let's talk about how it works. So the energy stored by this electron transport chain is something a little bit like water behind a dam, and I hope you guys are familiar with how this works, right? So I hope you have seen a dam before, but here's a picture, right? What we can't see is on the other side of this dam is probably a lake, right? Or maybe a big river, but probably a lake. Point is, there's a lot of water, and this dam is keeping that water back. And gravity makes the water want to move this way, right? That's what gravity does. But we have stopped it from doing that. So what happens is we let some of the water out, and as that water comes out, it moves a turbine, right? Assuming this is an electrical dam, it moves a turbine, and that turbine spins, and we produce electricity from that. And this is very similar to how the mitochondria works. So what happens here is we are pumping hydrogen ions from the inside here, right? This matrix, and I'm not going to test you on that word, but yeah, we have all these hydrogen ions on the inside of the mitochondria. So we pump them out using those proteins, and they come out here, right? That's a horrible drawing, excuse me. They all come out to this inner space, this space between the inner membrane and the outer membrane. So these electrons, again, they're going from protein to protein, and these proteins are embedded, and you can't see them in this picture, but they're embedded in the inner membrane. As they go from protein to protein, they are releasing that energy, and you can almost think of it as pumping water up to the other side of the dam, right? So the water does come down the dam naturally because of gravity, right? And that's how we make electricity. But we could also use electricity or some other mechanism to pump water against the flow of gravity. And that's what's happening here. We are pumping hydrogen ions against their concentration gradient. So this is where we go back to what you learned in lab last week, right? When we talk about diffusion, right? So let me see what that is. Excuse me. Sorry, guys. Good question. Yes. So, yes, we are pumping those hydrogen ions out of this inner area, and then they're trapped in between the inner membrane and the outer membrane. They're trapped there. There's a bunch of them. We're pushing them against their concentration gradient. It's highly concentrated hydrogen ions in there now. So what's going to happen? Like, as you should know by now from, again, lab last week, when there's an area of high concentration and an area of low concentration, diffusion is going to want to happen, right? So these hydrogen ions are not going to want to stay on the inside in between these membranes. They're going to want to diffuse back down to the inside of the mitochondria where the concentration is lower. So let's talk about that. It's the ATP synthase that allows them to do this. And there's going to be a picture. I'll draw it, or I'll show you a good picture later. Until then, I'll make this horrible drawing. Let's call that ATP synthase, right? We've got the outer membrane here, the inner membrane here, right? Then we have the inside of the mitochondria. Maybe I'll just finish it. There you go. Outer membrane, inner membrane. Anyway, again, we're pumping hydrogen ions against their concentration gradient. So there's a bunch of these H plus ions trapped in between the inner membrane and the outer membrane, and they want to diffuse back down, but they can't just go through the membrane. So they have to go through this enzyme called ATP synthase. They just diffuse right back down to where they want to be. But in the process of doing this, that's where we're making the ATP. The process of these hydrogen ions going down the concentration gradient, it literally spins. This enzyme has a thing that spins on it, just like it would a generator on a dam. It spins it, and that's where we make our ATP. So the test questions, a lot of test questions here. Which of the three stages pumps hydrogen ions against their concentration gradient? That would be the electron transport chain. Which of the three stages uses hydrogen ions diffusing down the concentration gradient to produce ATP? That would be the electron transport chain. Which of the three stages, big one. Which of the three stages uses ATP synthase, right? And that would be electron transport chain. So far in this discussion, I mean, I've mentioned an enzyme before, but this is the first time I've given you the name of an enzyme in this discussion, and that's the only one I'm going to teach you. So that is obviously important. You need to know the name of the enzyme that makes ATP. Is called ATP synthase. And like I mentioned earlier, when we first started talking about enzymes, I said they always end, as far as we're concerned, they always end in ACE, and they're always named after their substrate or their job. Like, what is it that they do, right? So ATP synthase synthesizes ATP, hence the name ATP synthase. So those are some test questions. Now, this horrible drawing I have, I'm going to show you a better drawing, and we're going to talk about it in more detail. But are there any questions before we do that? All right. So the next word for attendance is for you to tell me the name of the enzyme that makes ATP. All right. Then let's move forward. So here's a better picture. This picture also, by the way, is not from your textbook. This is from the old textbook, and I just kept it because it's, I think, a really good picture. So what we have here on the right side of the screen is an entire mitochondria, right? And then we kind of zoom in. This little box right here means we've zoomed in. So now we're looking at this one very small portion of the mitochondria. And what we're looking at here is, here's the inner membrane with all these proteins built in. Here's the outer membrane. And then there's this space in between. And we're zooming in even more to this little box. So what we see here is the inside of the mitochondria, where we have all these H plus ions, right? And then we have the space in between. What you can't see is the outer membrane. Somewhere out here is the outer membrane. And what's happening, like I mentioned earlier, we have all these high energy electrons that we stripped away from glucose. Those high energy electrons are taken to the electron transport chain by NADH and also FADH2. And I introduced you to that when we talked about the citric acid cycle. Anyway, those electrons come here and they go from protein to protein. And as they're going from protein to protein, all that energy they have, that extra energy, is released. And that released energy is used to pump hydrogen ions against their concentration gradient, right? So now we have a really high concentration of hydrogen ions up here and a very low concentration on the inside. So they just do what they naturally would, due to Brownian motion. They diffuse back down the concentration gradient through this enzyme called ATP synthase. And as they do that, this thing literally spins and it produces our ATP. And that's what we're after, right? ATP. This picture also has some other stuff that's worth mentioning or repeating, I guess, because I've already said it before, but here it is again. Electrons have to come from somewhere. This picture doesn't show that, but they were stripped away by these molecules, right? The NADH and FADH2. They were stripped away. The electrons have to go somewhere. Where do they go? Oxygen. Oxygen accepts it. And then when it does that, that then gives us a molecule of water. So again, test questions. Which of the three stages produces water? Electron transport chain. Which of the three stages uses oxygen? Electron transport chain. What molecule ultimately accepts the electrons? Oxygen. What molecules formed when the oxygen ultimately accepts the electrons? Water. Which of the three stages pumps hydrogen ions against their concentration gradient? Electron transport chain. Right, so all these things, there's a lot of test questions just in this one picture alone. And this is actually a little bit more detailed than you're going to need to know for the exam. Yes, technically there are 3 proteins in this step, and they are each different, and they are in a particular order. It's not random. And yes, technically NADH is the thing that's bringing the electrons to the first protein, and it's FADH2 that brings it to the second protein, but you do not need to know that. You don't need to know those details. You don't need to know that there's 3 proteins. You just need to know that there are proteins in this process and that the electrons go from protein to protein. And when they do that, they release their energy, and then those proteins pump hydrogen ions against their concentration gradient. This little skull and crossbones, this is a little indication that this is how you die. When you don't have oxygen, this is why it's deadly. Because without oxygen, your cells can't produce ATP, and that's actually what kills you. Anyway, you can look into this too for independent work if you want. Like I said, there's 3 proteins. I didn't even tell you the names of them. I didn't even tell you how they worked. So you can look into all of that for independent work if you want. What are the names of those proteins, and how was it that they pump hydrogen ions against their concentration gradient? Anyway, any questions about this slide? All right. So here's a number, like I mentioned earlier. I gave you this number previously in a previous lecture, and I said you need to know this number. So here I'm giving it to you again. Overall, cellular respiration generates about 32 molecules of ATP per molecule of glucose. And of course, if you look at different sources, might say something different, like usually 38 is another number you see a lot. So when you're memorizing this, just keep in mind it's in the 30s, right? So this is the number I'm going to test you on, 32. But as long as you can memorize the fact that it's in the 30s, then you're good. Because I'm not going to try to be tricky. Like you're in the multiple choice exam, your choices aren't going to be like 31 ATP, 32 ATP, 33 ATP, or 34 ATP, right? I'm not going to be that tricky. It's going to be like 30s and 40s and 50s and things like that. Anyway, any questions? All right. So here we go. So this is from the old textbook and not in your textbook. And I don't know, I guess I didn't even really necessarily need to include it, but we've already talked about all this. So again, this is just a good one picture to study. Um, so for example, right, if I ask which of the three stages produces ATP, they all do, right? They all produce ATP. But then I'll get a test question that says which of the three stages produces the most ATP? That would be the electron transport chain. Which of the three stages strips away high energy electrons? Like what's happening here, right? That's what's happening here. Which of the three stages does that? Citric acid cycle and glycolysis. Which of the three stages is associated with acetyl-CoA? Remember I said, as far as we're concerned, those things are together, right? Acetyl-CoA. Which of the three stages, of course, this isn't on here, but which of the three stages is associated with acetic acid? And that would be citric acid cycle. And again, if I could fix this, I would, because we don't call it pyruvic acid, right? To keep things easier for you, we call it pyruvate. So which of the three stages produces pyruvate? Glycolysis. Which of the three stages uses glucose? Glycolysis, right? A lot of test questions here. Anyway, one thing you don't need to know, like I mentioned earlier, yes, you do need to know we have about 32 ATP, but I'm not going to ask you how many ATP are produced in each individual step. You do, again, you need to know that it's electron transport chain that produces the most, and you do need to know that the other two produce a little bit, but I'm not going to ask you those numbers. Likewise here, like I'm not going to ask you how many molecules of NADH and FADH2, right? Those are questions I'm not going to ask you for the exam. So, any questions before we move forward? All right, then let's move forward. So, everything I told you, hopefully you remember this, is a big important concept. That's all because of oxygen, right? Like I said, all those electrons, those high energy electrons, have to go somewhere, and they go to the oxygen. So without oxygen, you don't have that, right? And remember that little skull and crossbones, because generally what happens without oxygen, at least for humans anyway, and most animals, or all animals, if you don't have the oxygen, eventually you die, right? But there are some exceptions to that, and that's what this next section is. Let's talk about fermentation. And for your homework, I expect you guys to drink a bottle of wine or a case of beer tonight. No, I'm just kidding. Please don't do that. But anyway, yes, fermentation is indeed, though, how we make alcohol. So let's talk about fermentation. Here's the learning objectives of this section. By the end, you should be able to discuss the fundamental difference between anaerobic cellular respiration and fermentation. So it's going to get a little trickier. And you should also be able to describe the type of fermentation that readily occurs in animal cells and the conditions that initiate that fermentation. So with that in mind, let's talk about these things. So again, you need oxygen, right? Our cells need oxygen to survive, but they can survive short periods without oxygen. And that's what fermentation is. Fermentation is the anaerobic harvest of food energy. So you should know that. Anaerobic harvest of food energy. So this is basically what happens. Again, it's the best that cellular respiration can do without oxygen. So you take away oxygen, those electrons don't have anywhere to go. So your cell's like, well, all right, we'll do the best we can. And the best we can is fermentation. So we're going to talk about it in more detail, but are there any questions so far? All right. So after functioning anaerobically for about 15 seconds, your muscle cells start to generate ATP by this process called fermentation. I'm not going to ask you that. I mean, your book talks about it, but I'm not going to get that detailed. So you just need to know that, hey, if you take away the electron or take away oxygen from our cells, not even muscle cells, I mean, technically that's what we're talking about, but you don't even need to know that much detail in the exam. When we're talking about just cells in general in our body, if you take away oxygen for a while there, they're gonna continue to make ATP, and they're gonna do it via this process called fermentation. One thing you definitely need to know about fermentation is this right here. It's only glycolysis. So we're no longer doing the entire electron transport chain. We're not doing the citric acid cycle. We're not doing Yeah, the electron transport chain, right? We're only doing glycolysis. And as you remember, glycolysis didn't produce that many ATPs. That's why this is a very temporary solution, at least for humans, because we can't survive on that alone. But yeah, the reason we use glycolysis and not the other two is because glycolysis does not directly require oxygen. Remember, we were just stripping away electrons from the glucose and producing pyruvate. And then those electrons went somewhere, right? Those FADH or NADH, right, took those electrons to the electron transport chain. But they didn't, the oxygen itself was in the electron transport chain. It was not in glycolysis. So glycolysis does not directly rely on oxygen. It does indirectly, and we're going to talk about that here in a second. But yeah, it doesn't directly require oxygen. So what happens is, the problem with the situation is we keep stripping away these high energy electrons, right? We use NAD plus to strip away the high energy electrons. And technically, once they get those electrons, it becomes NADH, right? And what used to happen, or what does happen in the process of oxygen, is those electrons are taken to the electron transport chain. They go down. Remember, they go from protein to protein until finally oxygen accepts them and becomes a molecule of water, right? But if we don't have that oxygen, then here we are stripping away these high energy electrons and we have nowhere to put them. And that's where the problem is. So this is where this is what basically drives fermentation, if you will. So again, here's just a visualization of what I just said, right? So usually this is what usually happens. We strip away these high energy electrons. They go to the electron transport chain, where they are ultimately accepted by an oxygen molecule, which produces a molecule of water. But if we don't have this, then this is what we're stuck with. We're stuck with all these NADHs that are filled with electrons, and they're filled, right? So they're not empty. They can't strip away any more electrons. So we have to find a place to put those electrons, because we no longer have the oxygen to dump them to. So this is what happens. And this is what you'll need to know for the exam. Since there's no oxygen for those NADH to drop their electrons off at, they instead put it back to the pyruvate. And to remind you, in glycolysis, we start with glucose, right? One molecule of glucose, and then we produce 2 molecules of pyruvate. Not that I'm going to ask you how many, but the point is we start with glucose, we end with pyruvate. So what happens is, while we're stripping away these electrons from the glucose, since we can't bring them over back to the oxygen like we do in aerobic situations, when we're without oxygen, then those electrons are just put right back to the pyruvate. And I'll show you a picture of that later. And that's what we need, right? So once those electrons are dumped off onto those pyruvates, then we have NAD plus again, right? So we have that empty electron acceptor, right? And it's dumped off its payload, so now it can strip away some more electrons, and that's what we need, right? So this is what keeps the glycolysis working, is those high energy electrons are being dumped back off onto the pyruvate. Now here's what you need to know. When we do that, this is what it produces: lactic acid. And I will admit that's a little bit tricky, because I told you earlier, for the exam, anything that I mentioned about acids, if it was about cellular respiration, you should think of the citric acid cycle, right? Whether it be citric acid or acetic acid, you should think of citric acid cycle. This is an exception, but technically we're not even talking about regular cellular respiration, right? We're talking about anaerobic respiration. So yes, you should associate lactic acid with fermentation in animal cells. I mean, there's other cells that do it too, but and I'll talk about that here in a second too. But yeah, that's how we produce lactic acid. This is why, for example, one of the reasons that your muscle cells will burn, like, right, if you're, I don't know, sprinting, right? If you're just doing a high energy workout and you're out of breath and your lungs burn too, this is why. It's because you are using more ATP and you're using so much oxygen, you need more of it, and your cells don't have a place to put those electrons. So they're putting it back onto the pyruvate instead, and that produces lactic acid. And that's what you need to know. Technically, this is also what happens. Technically, the lactic acid is then transported to the liver, where it is converted back to pyruvate. But I'm putting an X to that, indicating that I'm not going to ask you that much detail on the exam. But you do need to know, in humans, I mean, other things too, but I'm just sticking with us. In humans, this is what happens, right? When we're without oxygen, this is what our cells resort to. We're still stripping away high energy electrons, but we're putting them back onto the pyruvate. And the process of doing that produces this thing called lactic acid. So any questions about that? All right. And here's kind of a picture I was talking about, right? Like this person, I assume, just went for maybe a sprint and maybe their muscles are burning. And that's probably one of the reasons why, is because they're producing lactic acid. And here's a visualization of everything I said, right? So we're dealing with glycolysis here. So we're breaking glucose down into pyruvate, or I wish I could just fix that. Yeah, we're calling it pyruvate, not pyruvic acid. We're stripping down we're breaking down glucose and we're stripping away these high energy electrons. But since we don't have anywhere to put them, we put them back onto the pyruvate, and that produces lactic acid. And yes, technically there's 2 molecules produced. And yes, technically we're using 2 molecules of this. But I'm not going to get into that much detail. You just need to know that in fermentation in human muscle cells, we're using glycolysis only. We're breaking down glucose. We're stripping away electrons, and those electrons are put back onto the pyruvate, producing lactic acid. So the next word for attendance, let's see. When we strip away high energy electrons and put them back onto the pyruvate, then what molecule is formed? So I've said it. You saw it in writing in the previous slide. You see it on the picture here. So what molecule, or technically molecules, what molecule is produced when we put the electrons back onto the pyruvate? And now I'll move forward if I don't have any questions. If you guys don't have any questions. Well, it's 8:40. I don't have any announcement reminders slides, so nothing visual. But I guess just to remind you, you can still retake the first exam. As far as I know, I think only one person has taken me up on that offer, but you have nothing to lose. You just go in there and take it. You can retake it as many times as you want. It's not timed. It's open book. If somehow you keep getting a lower score than you started with, then we'll just keep, just don't mention anything to me and we'll just keep your higher score. So you have nothing to lose. Additionally, the first 3 exams are what I use to write the final exam. So in a sense, exam 1 is a study guide for the final exam. So even though you're done with the exam, you're not done with it, right? You still need to know that information. So yeah, I highly recommend doing that. That offer is open until it's time to take exam 2. At that point, you can no longer retake exam 1, but at that point, you can meet me and I can give you all the correct answers to the exam and, give you that feedback. So not only will you get all the correct answers to the exam, which you'll need when you're studying for the final exam, but also, again, you'll get a 5% boost to that grade, which is a good time to remind you that stands for anything as far as labs, exams, and quizzes go. I will meet you and give you detailed feedback on your assignment. And not only will you get the detailed feedback, but then just to incentivize you to do this, I will give you a 5% boost to that assignment's grade. I think that's it for announcements and reminders. Lab will be as usual. We're going to meet, 10 a.m. In the normal room on Wednesday. This time, though, just to remind you, we are not doing an experiment this week. We're going to do basically the pre-lab lecture. I think that's it. Yeah, that's it for announcements and reminders. And don't forget to write independent work as a whole letter grade. So please get it done. I can help you if you need ideas. Let me know. Any questions? Do you guys have any announcements? All right, so then let's move forward. So again, this is what happens in human muscle cells, right? This is how we produce lactic acid. This is how the process of fermentation produces lactic acid. If you download this PowerPoint, you can watch this little animation that kind of shows you what I just showed you, but animated. So hopefully it'll make it a little bit more sense. And this video, though, also talks about this. So there's 2 different types of fermentation. There's the one we just talked about, which produces lactic acid, aka lactate. And then there's another type that we're about to talk about that produces ethanol, which is just alcohol, right? The kind of alcohol you would drink in a beer or liquor or wine, whatever. It produces ethanol and carbon dioxide, and that is what we are about to talk about. So, again, for humans, when we go without oxygen, then for a while there, our muscles are just doing fermentation, and everything's kind of okay for a little bit because we're producing a little bit of ATP, but that will not sustain us, right? That's deadly for humans. We need to breathe oxygen, right? We need oxygen to survive. We're big organisms. However, there are some microorganisms that can live completely off of fermentation. And we take advantage of this to produce things like cheese and sour cream, yogurt, soy sauce, pickles, cabbage, olives, sausage, meat products, like any kind of cured meats, all these things. Not that I'm going to ask you, this is not a food course. Actually, it would be fun. We should have a food course about biology. But anyway, yeah, we use yeast, right? So we use this anaerobic respiration to produce lactic acid, and then yeast, uses that to make all these different products. And if you want to, you can look into that for independent work, right? What are some foods that are produced by yeast? And yeah, yeast is a microorganism. It is a single-celled organism. It's interesting because it's a fungus. So that means it's a eukaryote, just like us. Remember, a eukaryote, not a prokaryote. So it's more advanced, complex cells with organelles, but it is only single cellular, and it can produce, or it can live off of fermentation alone. But notice I'm talking about lactic acid still, right? I still haven't said anything about, I think, what most people associate with when they think of yeast and they think of fermentation. So we're getting there. So, like I said, they are single-celled organisms, but they're eukaryotes. So this is kind of what makes them interesting. They are capable of doing regular cellular respiration like we do, but they can also produce yeast, lactic acid, like we talked about in the previous slide, and they can do this completely other type of fermentation, which produces ethyl alcohol or ethanol, right? As far as you're concerned for the exam, if you just remember alcohol, you're fine. There are different types of alcohol, but I'm not going to ask you about that on the exam. So if you just remember that yeast uses fermentation to produce alcohol, you're good. It also happens to produce carbon dioxide in the process as a byproduct, and we'll talk about that here in the next slide. But yeah, this is why, for example, beer is carbonated, right? Because in the process of turning that glucose into alcohol, we've produced also carbon dioxide. And also, not that you need to know this again, this is not a cooking class. But yeah, this is why bread rises. Because when we make bread and there's yeast in the bread, the yeast is producing the ethyl alcohol, but it's also producing the carbon dioxide, right? And that carbon dioxide is literally gas, right? So these bubbles, and that's what makes it rise. But that does beg a question if you want to look it up for independent work. When you think about bread, right, it doesn't, if it was cooked properly, it doesn't have an alcohol taste, right? It doesn't have an alcohol flavor. You don't get drunk from eating bread, generally speaking. So the question is, if you want to look it up for independent work, is where does that alcohol go? And yes, it's not a cooking class, but that is biology related. So feel free to look that up. When you're making dough, when we're making bread, where does that carbon dioxide go? I guess you could say the same thing too for other stuff like wine, for example, right? We're using yeast to take that glucose that's in the grape juice and break it down and turn it into alcohol. And wine is not carbonated, right? If it was, it would be called champagne or sparkling wine. So where does the carbon dioxide go, right? So first I'm asking you, where does the alcohol in bread go? Now I'm asking you, where does the carbon dioxide in wine go? Anyway, any questions about this slide? All right. Here's a picture of how it works. This should look very familiar. It should look a lot like the other picture I showed you. Again, we start with this glucose, right? Because this is all happening in glycolysis. We start with glucose. We're breaking it down into pyruvate. We're stripping away high energy electrons. Those electrons have nowhere to go because this is an anaerobic situation, right? So there's no oxygen for us to dump those electrons at. So where do we dump them instead? We dump them back. But in this case, instead of dumping them back to pyruvate, It's not technically pyruvate anymore because in this process we lose the carbon, right? So technically we are then left with alcohol, ethyl alcohol, ethanol, however you want to call it. Just remember for the exam, if you just remember alcohol, you're going to be fine. But yeah, so that's how it works. And that's where the carbon dioxide comes from. Is in this process. Unlike the last one we talked about, when we talked about lactic acid fermentation, we didn't lose carbon dioxides, right? We just kept that whole entire 3-carbon pyruvate. We added the electrons to it, and that produced it. Produced it. Good grief! That produced lactic acid. In this case, we're not keeping the entire 3-carbon molecule. We're losing some carbons, and then that gives us again alcohol. So, any questions about this slide? All right, we've got about 2 minutes left. So it is also worth noting that some prokaryotes, like I mentioned earlier, prokaryotes are basically just bacteria. As far as we're concerned, you can just consider them bacteria. There are some other things, but it's just easy enough to think of prokaryotes as bacteria. They are able to use anaerobic respiration in different ways. So you have things called methanogens, which reduce carbon dioxide to make methane. And here's a term I haven't taught you yet. You don't need to know this for the exam, but when you're reading the textbook or anything else, oxidizing something means that's taking away their electrons. So when we say we're oxidizing NADH, that what I mean by that is, like I said previously, we need to get rid of those electrons, right? So that then we can have NAD plus again, right? So because when it's filled with electrons, it's no use to us. So we need to get rid of them. And yeah, methanogens can do that. There's also sulfate reducing microorganisms that turn sulfate to hydrogen sulfide. But guess what? You don't need to know any of that. It was in the book, so I included it. So when you're reading the textbook, you won't say, what? He never said anything about this. So there you go. That is the end of that discussion. It is now 8:50. I guess that's not the end of that discussion, but we'll come back to it. So not the last word for attendance is not a word at all. Once again, as usual, send me a question about the material that I presented. Even if you think you understood everything, just send a question just to make sure. And that's it. I will be online until 10 a.m. for office hours, and I'll see you on Wednesday. And don't forget to send the words, please. Just because you're logged in does not mean